BPS Forensic Laboratory, Santa Mexico. Welcome, ma'am. Now, I request our energetic HOD, Dr. R. Devika, HOD Biotechnology Department, Arbada Video Institute of Technology, to present a welcome address. Please, ma'am.
Thank you, sir. Good morning to one and all. It gives me immense pleasure in welcome you all for the concluding session of the international webinar series on trending technologies. It is a great honor for me to welcome a very senior and eminent educationalist and a scientist, Dr. Abhirami Chidambaram, Quality Assurance Manager, NMDPS, Forensic Laboratory, Mexico. Ma'am, on behalf of our management, campus director, principal, vice principals, heads of all the departments, and on my own behalf, I welcome you, ma'am. I am indeed glad to welcome our senior most and young energetic person of AVAT, Dr. Vijendra Babu, our vice principal part-time studies. Welcome you, sir.
Thank you.
I am very much happy to welcome our dynamic and beloved Dr. Giri Rangaswamy, Dean CSR VMRF. Ma'am, I take this opportunity to thank you for your support throughout this webinar series and guided us to succeed in our efforts. Welcome you, ma'am. I take this immense pleasure to welcome all the participants, faculty members, student participants for this concluding session and for your active participation throughout this webinar. Welcome you all. Thank you.
Thank you, ma'am. Now, I request our beloved Vice Principal, Part-Time Studies, Dr. D. Vijendra Babu, sir, to give a special address. Please, sir.
Thank you, Dr. Madhusudan, sir. Good morning to all of you. Special welcome to our chief guest of today's event, Dr. Abhirami Chidambaram, madam. Welcome, madam, to AVAT for accepting your invitation on your busy schedule. And we are happy that you are addressing on the significant topic, human identity testing and forensic investigation, organized by Department of Biotechnology. A warm welcome to you on behalf of AVAT VMRF, madam. Good morning to Dr. Giri Rangaswamy, madam, Senior Faculty Member in Department of Biotechnology, Dr. Devika, madam, HOD, organizers, and the faculty members of biotechnology, and the participants from various industries, organizations, and across globe. Also, a special welcome to all our students who are in assembled here. It is my indeed pleasure in participating in the event organized by Department of Biotechnology, as they have been organizing various events. This is one of the key event as an international webinar series on trending technologies. As we all know, biotechnology is not a separate field or medicine or oriented or pharma oriented. It is a necessity area which now everybody has started focusing it. Whenever there is a trouble, we see always for a solution. In current pandemic situation, now everybody is expecting what is the vaccine next? How can I get out of from this issue? What is the corona? How it is going to be pandemic situation? If corona goes out, what is the next some virus coming from some peaks in China? So all those things are getting a fearness in world actually. So now all the focus is on pharmaceutical sector and all those things, food processing and health sector. Health sector now plays a major role as Department of Biotechnology in our institution as a leading department in wide variety of huge infrastructures, and they had been appreciated by Government of India by receiving grants for various projects, and the faculties are all doctorates and deeply involved in research activities. And I take this opportunity to congratulate the entire department and Dr. Devika madam for releasing, and it's going to happen. They are going to release a book which is going to be published on account of various faculty members on this pandemic situation. So that is a commitment which the department is actively involving with collaboration of all those departments. As rightly as said, this days, 5 days, is be of several topics of significant topics, like with respect to various diversified areas. As maybe it may be so technical, but as an end user, it is going to for the betterment of humanity. It's going to the betterment of society. It's going to be the better societal life or whatever thing. Even the WHO or all those factors now focusing on health sector. What is going to be next? Which is good for us? What is not to be taken? What is the social lifestyle? All those things. I hope this 5 days event of international webinar series is going to be a key takeaway for all the members who have been participating. And I really wish the organizers and especially Dr. Devika Madam, HOD, Biotechnology, and the entire team who has been supporting her for making this to happen. Once again, I wish the team for the best success and thanks for the opportunity. Thank you all.
Thank you,
Thank
sir.
you very much.
Thank you, sir. Thank you very much, sir. Now I request our dynamic Dean, Dr. Giri Rangasamy, Dean CSR, CSF VMRF, to give a special address. Please, ma'am.
Good morning, everyone. Today is the concluding session of our webinar series, which started on the 29th of June. Now, a few words about Dr. Abhirami Chidambaram. I feel very proud to say that she was one of my earlier students who did botany in Women's Christian College. She was an outstanding student and she was the only first class that year. And then I know she will make it big because her inquisitive mind and then going in here and there to do some reference work. She was a very vibrant and active student. Then she went on for her higher studies to States, and her PhD itself is from Maryland. Baltimore, it is on DNA. And though several years have gone after she finished her BSc, we were in touch. And I'm sure it is about our association is about 40 years, I think. And then whenever she lands up in Chennai, I used to make her talk because she was doing something very, very current and very, very important to life. So she has spoken in all the platforms in most of the colleges where I have friends. And you'll be surprised to know that she has come here twice. And she even went to Pondicherry in Karaikal to talk about DNA and forensic medicine. And today I'm very happy that she is here with us to share her knowledge, because she was from one level to another level she was going. Now she's in the highest level. And she even told me that during Rajiv Gandhi's murder, they gave her the sample to finger type, and she did the DNA fingerprinting. And today, the moment Dr. Devika approached her, she said yes. So this is the advantage of having students who are spread all over, especially my students who are still attached to me. I'm thankful to God for that. Abhi, and I'm sure what you are going to talk will be something new to all of us. And I'm happy that you could share your views with us. Okay. Now, I didn't know exactly what she was going to talk, because right now she said 3-4 years back she was even doing cyber crime, cyber security. She was originally working in the International Department of Forensic Science and Criminology at Alaska Anchorage. So today I thought I will just say a few words about cyber crime and security, so that the participants can have an idea, though it is a bit deviated from what she is going to talk. Online systems create a cyberspace within which people can communicate with one another, do research, or simply window shop. The physical cyberspace contains objects like files, messages, graphics, etc., and different modes of transportation and delivery. Cyberspace does not require any physical movement other than the pressing of the keys or moving the mouse. As a social experience, individuals can interact, exchange ideas, share information, provide social support, conduct business deals, create artistic media, play games, and so on and so forth, using global network. Thus, cyberspace is an online world with an environment for online computing. It is the futuristic online world. In this age of information explosion, technology touches every aspect of our daily life in some form or the other. The TV that brings us the morning news, the internet that delivers our emails, or the cell phone we use to make phone calls, etc. Through these wonderful inventions, we keep in touch with our family and friends, have business deals, all within the touch of a button, um, saving time and money. Thus, technology provides numerous advantages which were not available 35 years ago. But we should realize that there are disadvantages too, like a great deal of time may be wasted by addiction and even alienating our own families and friends. The children could handle inappropriate materials. Gambling may promote the use of alcohol, ganja, etc. Chatting during odd hours in the night, alluring children, etc. etc. The mobile phones can be used for destructive purposes also, and they are quite dangerous too. Teenagers communicate through phones and negative activities can follow. So these are some areas where cyber crime can can come in. And now, Abhi, all our colleges are going into e-learning, digital learning. So they say if it is a case, then it is bound to have a number of malpractices. So cyber security is very important. And I'm sure that our staff are getting prepared for online teaching. That will be the in thing now. And educationists say that two by three portions will be online and 1/3 will be inside campus. So they have coined a new term, phygital, that is physical and digital. So that will be the current topic now. And now I would like to thank Abhirami. So I'll hand it over to her.
Thank you very much, ma'am. Thank you for your elaborate speech and welcoming our guest, ma'am. Now, I take the immense pleasure to introduce our today's guest speaker, Dr. Abhirami Chidambaram. Dr. Abhirami Chidambaram, presently working as Quality Assurance Manager, Forensic Laboratory Bureau, New Mexico, Department of Public Safety. She has completed her UG from University of Madras during 1975 and B.Ed from University of Madras in 1980. And she completed her PhD in Human Genetics, GSPH, University of Pittsburgh. And she is having a teaching experience as adjunct professor, University of Alaska, Anchorage during 2000-2001 in the Department of Anatomy and Physiology. And she is also having experience in junior college, graduate school, training the forensic teams also. She has experience as DNA forensic biology, forensic analysis, and DNA technical manager, scientific crime detection laboratory, Alaska Department of Public Safety. And she served as a scientist in National Cancer Institute, FCRDC, Frederick MD, during the year 1993 to 98. And in 1991 to January 1993, she is a postdoctoral candidate in UTMD Anderson Cancer Center, Houston, Texas. And she also having experience as assessor in ISO 17025 forensic laboratory testing assessor since 2009. She is also expertise in DNA forensic biology, casework, and ODIS ISO IEC 17025. And she is also having a courtroom experience as expert witness in genetics, population statistics, more than 100 plus times. And she is also well versed in STR analysis, data interpretation, case management, grand jury and jury trials, SOA, rebuttal, expert witness, DNA admissibility, Robert hearing, sentencing hearing, all these things. And she also having a professional affiliation in American Society of Human Genetics, American Academy of Forensic Sciences, and Association of Forensic Quality Assurance Manager, AFQAM. And she has more than 40 international publications, and she is also having experience in DNA overview and sexual assault response team training for more than 20 cases. And she is also acted as invited speaker in various international conferences throughout the world. Dr. Chidambaram has experience in training for more than 50 companies in different areas of forensic sciences. Now I welcome on behalf of AVAT to present your speech, ma'am. Please, ma'am.
Abhi, you can start now.
Ma'am, I think Avi ma'am has been disconnected.
Yeah, you came in? Okay,
Yeah.
thank you. Are you there, ma'am?
Yes, ma'am. Ma'am has joined. Ma'am.
Okay, okay.
Can you see me now?
Video, ma'am.
Start your video, ma'am.
Yes, there we go. The video has been started.
But we cannot see you.
Can you see me now?
Yes, ma'am.
Yes.
Yes.
Okay.
We can hear you also.
All right. Yeah, we in the middle of your talk, Dr. Giri, the connection just went off, and I've been trying to reconnect since that time.
Sorry.
It's okay. Okay. So I don't know where we left off.
You have
Welcome, ma'am.
it's
Now
your
the session is yours,
right
ma'am.
now, ma'am. Sam, you can share the screen.
Yes.
Yeah, start now, Abhi.
Okay. And can I have the, okay, there we go. Thank you so much.
Okay.
So thank you, sir. Yeah, I don't know. I hope it doesn't get disconnected again. So
Confidently, you can go on.
thank you. So what I wanted to talk about today was about the technologies, but it's an extremely applied science, the way another person here just explained, because there are so many different ways. It's not pure science, but it's the application of all the basic sciences. And the other thing I wanted to clarify is it's New Mexico, and I'm not in Mexico. I'm still in the United States. It's the state of New Mexico. That's where my lab is located. Okay, so coming back to the talk, we are only going to focus on human identity testing and forensic investigations. So this is even a narrower scope of all the technologies that we use. Oops, okay. So it's used, human identity testing is used for forensic casework, paternity testing. Paternity testing can be a civil case or a forensic case. History and anthropology investigations, like, there's been some questions that have come up in the course of history, for example, as going back as far back as 6,000 years to the mummies. They are trying to see paternity situations with King Tutankhamun. And then in history, there are a lot of situations that have come up where people have said, oh, I'm related to this president or that president. This is the kind of investigations that are done. Again, there are missing persons investigations, mass disasters. There was Katrina, the tsunami in 2004 that hit India and a lot of places in that part of the world. And then the World Trade Center, where we don't find whole human bodies, and they are, I mean, very distressing, but they can be only collected in bits and pieces. And this is the kind of testing that puts all the pieces back together. And then military people in the armed forces, when they go out to fight and they're missing in action, when they find the remains, maybe after 10-20 years or sometimes 30 years or more, there is not a whole lot left to identify people. It's DNA that's used to identify them. So when people talk about identity testing, I don't want people to just imagine that it's used only for forensics and catching the bad guy or a gal and putting them in jail. It's used for a number of different reasons. So And educational background for doing forensics is actually very varied, and the more information you have, the more background you have is important. So for example, FBI, the forensic for the United States. They are the people who control the quality assurance and what needs to be done in this field. So they actually mandate or make it compulsory for people coming into this field to have a minimum amount of coursework and minimum amount of hours and semesters in certain areas. You have to be familiar with the classical Mendelian genetics. How do traits get passed from generation to generation? You have to be able to know something about biochemistry. You need to know recombinant DNA techniques, molecular biology. You have to have probability and statistics. This becomes very important. Statistically, the statistics become very important when you are saying, oh, this DNA profile matches this DNA profile, and say, let's say we are identifying somebody with this information. How much statistics do you need to be absolutely sure you're not sending the wrong person to jail or sending the right person off on the street and freeing somebody? So you need to be absolutely familiar with how probability works and how the statistical strength of the work that you are doing is to present in court. And then you need to know population genetics, because sometimes the probability and statistics are not the same across the board. Indians have a different statistical, or Asians, for instance, we have different markers in our genes, and that may be different from a Native Americans, or it could be different from a Northern Europeans, or from the native populations in South America. It's quite different when you have to show statistical significance when you see and make these matches. And it's also useful to know some physics and analytical chemistry, because you're going to be using computational algorithms to predict
Devika? Devika? Hello?
Sham.
What's happening? Sorry. For the previous slide.
Abhi ma'am?
Abhi?
I think ma'am got disconnected again.
Disconnected. Yes.
Participants, kindly hold on. Ma'am will be joining shortly.
Sir, Devika?
Giri ma'am, can you contact Abhi ma'am?
But what should I do? Tell me.
Can you just call Abhi ma'am to join, ma'am?
Yeah, I am calling her. Abhi? Abhi, can you hear me?
Ma'am, over the phone, ma'am. Over the phone, I mean.
Phone. Okay. Phone. I can call. Hello, Abhi. Why it got disconnected? Yeah. They asked me to call you. Yeah, it got disconnected. And now, can you call Devika? Any one of the panelists? Can you call? Sure. I'm just trying to join the meeting again. Yeah. So they asked me to contact you through phone. That's why I'm talking. Okay. So what was the last thing you heard me say? That in that list of physical and analytic or something like that. Last but one. Because I was talking for a while. I heard Devika, but yeah. Now it just says I'm connecting. Okay. Because I think that person is trying to connect, I think. Okay, now can you call Devika? Okay, okay. I'm also trying to connect. Okay, okay.
Update 1. 1. If
Devika?
Ma'am, Devika ma'am is also facing some technical issues, ma'am.
Jeshran, sir.
Yes,
Jeshran
ma'am.
Samuel, sir.
Yes, ma'am.
You are giving the connection, isn't it?
Yes, ma'am. Yes, ma'am.
Now you are trying for Abhi's connection.
Yes, ma'am. I'm
So
waiting to
all these
ma'am
days
to join.
it was very good. Only today this has happened.
decreasing of the
Yes. ஹலோ தேவிகா, தேவிகா
டிஸ்கனெக்ட் ஆயிட்டாங்க.
எப்படி?
தெரியல, அவங்களுக்கு ஏதோ அங்க ப்ராப்ளம் இருக்கும்ல.
அவள்கிட்ட நான் இல்ல, அவள்கிட்ட நான் பேசிட்டேன். அவ சொன்னா ஒண்ணுமே இல்ல, நீங்க சடனா எனக்கு நின்றுச்சு, அதனால யூ ட்ரை டு.
இப்ப ட்ரை பண்ண சொல்லிருக்கேன் திருப்பியும். பார்க்கலாம் வராங்களான்னு பார்க்கலாம்.
சரி, ஒரு நாள் இப்படி நடந்தது இல்லையே.
அதுதான் என்ன தெரியல, திருஷ்டியாட்டம் இருக்கு.
ஆமா, திருஷ்டி தான். அவ என்ன சொன்னான்னா, அவ பாட்டுல ஒண்ணுமே தப்பே இல்ல, அழகா போயிட்டு இருந்துச்சு, அண்ட் இட் ஸ்டாப்டுன்னு சொல்றா. என்னன்னு தெரியலையே.
அவங்கதான் இப்ப டிஸ்கனெக்ட் ஆயிருக்காங்க.
ஆமா, அவதான் இருக்கா, அதனாலதான் நமக்கு ஒண்ணும் கேட்கல. அதான் அவ லாஸ்ட் ஸ்லைடு என்னதுன்னு சொன்னா. நான் சொன்னேன் லாஸ்ட் ஸ்லைடு என்னன்னு. ஒண்ணு அப்ப அவ வந்து நான் பாக்குறேன் அப்படின்னா.
இவங்க ட்ரை பண்ணிட்டு இருக்காங்க போல, வரட்டும்.
சரி சரி.
I think I am finally.
Yeah, you're there, ma'am.
Yeah.
I'm
trying
video. You can maybe you avoid video, ma'am. You just go with the audio alone.
There we go.
Ma'am?
Yeah. I don't know what's happening because we occasionally have problems, but not this often. So anyway, I'll kind of speed through the talk a little bit. And do you think, okay, so I already did this. And so I was just trying to, I was just trying to impress on the fact that, impress on the fact that this area is so, it is so applied that you need to do a lot of, so you need all the background. So to go on with this kind of tech, do you think if I switch off my video, it might not crash?
Yes, ma'am. That's what I felt. You go out of video, ma'am.
Okay.
Maybe there's a problem there.
Okay.
You just go. Yes, ma'am. Continue, ma'am.
Okay. So let's talk about forensic cases. It's relatively simple, and then it's simple in the sense that we are trying to link all the DNA from suspect to victim and victim to scene and scene to object. But as you can see, the DNA might not be very fresh, and you may have extremely small amounts of DNA. And so you are going to notice from the technologies that I used that we are going to really enhance the technologies to suit the fact that we are not really getting good quality DNA. We may not even get a lot of DNA in most cases. So how do we get around this? And the DNA may be also degraded. So the technology takes into account all these problems when you are coming up with a system to look for the evidence, in which case, so usually there's a why are we doing it. This is basically we are trying to identify some people. Where are we getting the DNA from? It's from the biological tissues in the body. It could be from a suspect, it could be from a historical figure, it could be from a victim. Sometimes it could be just the DNA left on the tip of or the lip of a can, like a beer can or a soda can or something. If somebody takes a little sip, there's enough DNA there to do the testing. And then we need to know what is DNA and how does it work. And now we are going, but in this talk, we are going to focus on the hows. How are we going to use the technology, and how are we going to improve the quality of the results we get from DNA that might not be the best DNA to do our research with or our testing with. So the next slide, we talk about the blood, semen, saliva, urine, the root of the hair, teeth, bone, and tissue. All of these things have DNA in those. Like, for example, in the tooth, it's a really good source of DNA, especially in old and degraded human remains. For example, the tooth was what they used when they were looking at anthropological studies with Egyptian mummies, because the whole body is mummified, the DNA has been dried, and it may not be suitable for analysis. However, the pulp of the tooth contains a lot of DNA, and it is really preserved by the enamel and everything else that surrounds the pulp, and the bacteria cannot get to it that easily. So, for example, bone is a tooth is a really fantastic way of getting DNA from really old samples. However, the way you have to get the DNA is not that easy. You basically have to grind it up with a drill, and there's contamination and all that going on. So there's different technologies for getting DNA from each of these different types of samples. Even though you're getting the same DNA from each of them, the techniques that you use for recovering the DNA can be very different depending on the tissue from which you are getting the DNA from. So this is the basic thing. It's a blueprint. So we are looking at all the autosomes as well as the X and Y chromosomes. And one of the tests that we use is a presumptive test for presence of blood. Now, we know that there is no DNA in a red blood cell. However, if you test for the presumptive presence of the hemoglobin in a red blood cell, even though the DNA might not be coming from the red blood cell, this positive test is telling you it's a possibility that there was blood here. So let's take a sample and test it. In fact, there was a very old case. There was a 30-year-old murder case where we really found the presence. We couldn't find anything else. And it did test positive for the presence of heme. And we managed to get a partial profile from the person who had been dead for nearly 30 years. So it's possible sometimes. Sometimes there is luck involved, but also the techniques. So here we have a set of human female chromosomes. So the chromosomes from the first pair to the 22nd pair, they're all going to be the same. But in a female, you got two X's, and in a male, you have an X and a Y. So pretty much the markers that we use for looking at DNA in human identification are spread across these chromosomes, and also you have loci which are specific to the X chromosome and which are also specific to the Y chromosome. That is a relatively new technological trend where we are trying to get haplotypes and haplogroups from the Y chromosome, which can help us identify to some extent what the paternal lineage of the person was. So it's obviously going to work only with males. And Occasionally, you can also tell what population or ethnic background the person came from, depending on the haplotypes. So there's a lot of work that's going on. In fact, if you go to the website, on the internet, you have something called YHRD. It's a searchable program, and you can look at all the background for a lot of different people. So here are 13 chromosomes, 13 loci that we were using in forensics until a few years ago. Now, the core loci, which you can see on the chromosomes, they are spread across all the autosomes as well as the sex chromosomes. So there used to be 13 in the past, and it was considered sufficient for statistical significance. But now we have gone from the 13 core loci to 23 core loci. So, and also we are adding all these chromosomal locations. Loci on the Y chromosome. So you have a bunch of them, and most of them are linked. So they are haplogroups, and they are not going to be separate, but these are the ones that help us analyze both the paternal lineage of the DNA that you're testing, as well as, if you're lucky, you can also find out what their ethnic background was. Now, with the good old days, and I'm talking about 1999, when I started in forensics, Profiler Plus had 9 loci. And if you look at the top line here, 100 base pairs, 200 base pairs, 300 to 400. 400 is pretty much the maximum size of an amplified product you can expect to find sometimes from a forensic sample. And so the primers are designed. So each one of these is a primer, and they're color coded. We no longer use radioactivity. These are all fluorescent primers, and the fluorescent tags are attached to the primers. So you can look all the blue loci, the green loci, and the yellow loci. So there were just 9 there. Then they added a few more to bring it up to 13. And at the bottom, you see these are the sizing standards. And at the top, you say this is going to be the maximum size of your DNA. And there's a good reason for that. In most of the cases that come for DNA, the DNA is not fresh. It could be degraded, but there is a good chance that these primers are pretty robust to find these fragments, which are at least about 400 base pairs. That's not a big size, but still it's something. So then one of the companies called ABI, and now they're called Thermo Fisher, I believe. And these are all global companies, so you may be familiar with some of them, although some of the loci that I used in different countries are different. So the US uses certain loci, and Europe uses some other loci, and India might use some other loci. I'm not 100% sure what the exact loci they use. I'm familiar with the ones that they use in the US. But if there is an international case, they actually pool their resources together, and there are many loci which are common globally, and some matches can be made. So again, even when you look at the identifier locus, it starts with 100 base pairs, and pretty much the largest fragment size you're going to get is going to be 400 base pairs or less. And PowerPlex 16 is a kit that's made by another company, which is called Fromega, and they came up with another kit, and that was looking at 15 location or loci. And they have they have 3 loci, which actually are slightly larger than 400 base pairs. Now, if the DNA is good, they work. If the DNA is not in good condition, it's not going to work. But still, even without those 3 loci, you're going to have reasonable statistical significance with the remaining loci. And so this is how all the possible alleles that are there at each locus look like. And this is the algorithm that the analytical software uses. So when we send a DNA sample in for analysis after amplification, you are going to get at least one or two of each of these locations. And each of the peaks in each of these boxes represents the number of alleles that can be found at each of those chromosomal locations. So you are looking at, so if you're getting one peak, one allele from dad and one allele from the mother, you are looking at, well, if you're heterozygous, you're going to be looking at two alleles. And if you're homozygous for the same allele, you're going to see one peak. And that peak is going to be twice the size of the two smaller peaks. So in a sense, at the bottom, the analysis of the DNA that we are trying to test is being sized by an internal running standard, which is run with all the samples. And then you can also compare it with all the variations and all the potentially possible alleles that are located at each of those locations or loci. Now, this is just, we are looking at just 13 loci here, and we have gone now to 23 loci, and it can look quite crowded, and you have to have large printouts. Now, in the best case scenario, and I'm going fast here because I don't want the system to crash again. So if you're looking at this profile, you can look, this is a clean single source profile. This DNA came from just one person. And if you look at this location that I'm pointing to here with the mouse, it says X and Y. So what it means is this sample came from a male because it has an X chromosome and a Y chromosome. Now, if this had come from a female, you would see only a X peak, and that would be a little bit taller than these peaks. Now, this is the simplest case scenario, and you have the sizing standard at the bottom. It tells you what the base pair size of each of the fragments are, and each fragment is labeled based on the ladder information that we gave. So it goes back and forth. So you're looking at three things are happening here. The fragment that came out of your testing is being compared to the size of the ladder at the bottom, and then it's being compared with the size of the alleles in each locus. So that is how we come up with a profile. Now, that can be a nightmarish situation where the DNA came from more than one person, which is not going to look as clean. And now you're going to look at a profile which looks like this. Some peaks are small, some peaks are really tiny, like this one. You don't know if it's a real peak or if it's just dirt in the machine. And so this is a DNA mixture. These are extremely difficult to interpret because we don't know how much DNA was contributed by each of the people in this mixture. If you look at this, there are at least, you don't know whether there are 3 people or if there are 4 people or if some people are missing from this mixture. So in the past, it was always been difficult to go to court and talk about these kind of mixtures and say, oh, we don't know who's in it. So again, technology to the rescue. So this happened maybe about 15 years ago. There was this person actually in Pittsburgh called Mark Perlin, and he came up with this program that looks at all the information in a mixture. He runs a company called Cybergenetics, and he has a software called True Allele. Now, if you send him your results, or you can buy the package from him, and you can run your samples, you can run the mixture, and it looks at everything from the bottom line to the top, and it gives you statistical probabilities of who's in that mixture and who's not in that mixture. So this has been a huge leap forward in processing those difficult to interpret mixture samples. Now, just last year, and this is something that we are even thinking of getting into our lab, this is called probabilistic genotyping software. This is also being taken care of, and this is called STR Mix. And it does the same thing that the other cybergenetics True Allele does, but this is something that we can run on the computers ourselves. And it is not as expensive as some of the other products which are out there. And it is designed to run on an individual DNA analyst PC that rather than having a different server or if you have a networked environment. and it doesn't need high-speed computing. It doesn't need fancy software. It just looks at mixed DNA profiles and does this probability and statistics calculations and comes up with similar results to what you got with the other software packages like TrueAllele, for instance. So these are some of the big steps that have happened within the last 20 years that I've been in forensics. So it seemed like change was very hard to come by early on, but now once change starts, it just goes in leaps and bounds. So now we have challenge samples, and they can be very challenging. So you are dealing with two things: inhibition versus degradation. So we get DNA samples in forensics sometimes from somebody's lips. And if it's a woman's lips, you may have lipstick combined with the DNA sample. Now, that lipstick and the fat in that lipstick can inhibit how your DNA works or how the DNA testing works. Or sometimes, in the case of human remains that have been lying out somewhere and exposed to the elements for a long time, there can be degradation. Now, there are different ways. The technology actually has improved to a point. It takes care of the inhibition and degradation too. And so for the degradation part, this company, which is Thermo Fisher Applied Biosystems, they came up with a kit called a Minifiler. Already we are looking at product sizes between 100 base pairs and 400 base pairs. They came up with a kit that would have fragment sizes that were amplified, which were less than 300 base pairs. So this was a great kit to use if you thought your sample was either degraded or if it was inhibited by any of the other items that might have been used. For example, if a killer decides to clean up the mess and uses a bleach or something, that can degrade the DNA. And so Minifiler to the rescue. And we actually managed to solve a few cases using Minifiler. So this is what happens. So you have fragments which are less than 400 bases as amplicons, and you're going to be able to look at those and get some kind of get some kind of information. Even if you feel you can exclude somebody, that is good information, because it doesn't have to be an inclusion or a match all the time. A mismatch or a exclusion is just as equally important in case you want to exonerate a person who has been wrongfully called in as a suspect. So all we do is now we have robots. There's not a whole lot of manual work that is being done. Now, extraction can be done by, for example, Kaizen. There are biorobots, and you just put these samples in these little cartridges, and it goes in, it comes out, and you have clean DNA. It cleans the DNA also for you. Then you can quantitate the DNA by using real-time quantitative PCR. So there are many kits for this, and there are many instruments for this. So once again, now the kit was used only for autosomal DNA. Now we have kits for YSTRs also. For example, this is a very expensive type of testing. The kits are very expensive. They don't have a very long shelf life. So if it's a sexual assault case, for example, and do we want to waste our resources by testing all the samples if there is no male DNA in it? So what happens is you use the YSTR loci to see if there is any, and quantitate and see if there is any male DNA in that mix. If there is no male DNA, the testing stops at that point. Again, the technology has evolved to a point where we are enhancing not only the sensitivity of the testing, but also saving resources and time. Because in many of these cases, the backlog is huge, and you have to be able to work as many cases without wasting resources. And then you amplify it using a thermal cycler. We use stack polymerase, which is commonly used, and then there are many kits for this. You collect the data, and there's genetic analyzer, which is used again, and there is software to interpret the data. Once again, pretty much everything has been automated at this point. So PCR, polymerase chain reaction, or PCR, has been the biggest boon in this area because it makes the amplification, which is like Xeroxing DNA, like a billion times, has come to the aid when you start with an extremely small amount of DNA and you have billions of copies to actually visualize and test and draw conclusions from. So I'm not going into all the details because they're the basic details. So this is a quote from one of the, so technology is increasingly able through amplification to take very small amount of found DNA and create a usable amount for testing. Sometimes it's an incomplete profile in a sample, and sometimes in very faint amounts that defy meaningful measurement. Sometimes more than one person's DNA is there. However, PCR amplification actually allows us to visualize the smallest amount of DNA in a sample. The only thing is, how do we interpret what we see? And now we have software we talked about earlier, like StarMix and TrueAllele, which comes to the rescue because it helps us. It takes the human error out of it. It takes the human I mean, the time, when humans do it manually, it's time consuming, but when a software runs it and they run so many computerized reiterations, it takes a fraction of the time to complete it. So in the old days, the ABI machine, the genetic analyzer used to look like this. This is the easiest to explain. So there's a heat plate and there's a capillary, and the samples go through the capillary. It's a capillary electrophoresis. It used to take about an hour to get a sample through each of those one capillaries. Now you have capillary systems which have about 24 that can be run simultaneously. So there's absolutely no bottleneck in actually the number of samples that can be analyzed. There are some industrial strength instruments which also have 96 capillaries. But for the lab, since the resources, the bottleneck for the lab is that you have to do review of the data, and it takes a long time. So we use either 16 capillaries or 24 capillaries, but all the same, the principle is the same. It goes through the capillary, and there's a camera that takes pictures, and the fluorescent dyes the fluorescent dyes are excited by laser beams going through the area, and then the camera captures it and you see the data in the form of the electropherograms. So coming to the more applications here. So that's the science part of it, and it's getting fine-tuned every day as we speak. And so, of course, there's nuclear DNA and you have mitochondrial DNA, and conventional wisdom says mitochondrial DNA comes only from the mothers. So, and for most part, that's true. And I say for most part because we have come across at least one example where that wasn't quite the case. So that's the other reason why you need as much background as possible, because when you see something that is not explainable, you need to find out whether it was just a mistake or whether there is something happening at the biological level. So look at the types of investigations. These are the few examples: forensic testing, maternal lineage, paternal lineage, historical investigations. For example, the remains of the Romanovs, and I'll give you an example of that. And I'll tell you how sensitive the testing was. And then Anastasia was a very famous story because this lady walked around most of Europe and she finally settled down in New Jersey in the United States, and said she was related to the czars, the last czar of Russia, and she was the daughter who escaped mass murder. And was she Anastasia? That mystery has been solved because of DNA testing. So, and DNA testing, as I mentioned earlier, is also used in testing 6,000-year-old mummy samples. And we get reasonably, or when I say we, the field, or people who do this kind of analysis, they get reasonably good data to do comparisons. And so coming to a historical situation here in the US, Jefferson, who was one of the early presidents in this country, he was a slave owner. And after his wife died, they had a governess who took care of his children in the household, and it's believed that he fathered some children through her. Her name was Sally Hemings, and she was an African American slave girl. And she had a number of children. And at that time, it was common knowledge. And then, many generations later, after all these things were said and done, a few people from the southern states came up and they said, "We are Jefferson's descendants, and we want some kind of a settlement." So this whole study came about at that point because these are all African Americans living in the southern part of the state, and they said, we can prove that we are all descendants of Sally Hemings, and we were fathered by Jefferson, so we are related to Jefferson and he's our ancestor. So they did YSTR testing, and they actually found out that he had a very rare type of Y chromosome, which was only present in European males. And they actually had some kind of a settlement made to these families in the South who are African Americans, because they could actually prove through DNA and the ethnic background of the Y haplotype that was found in Jefferson's male relatives. So that is the power of the testing when it comes to some of these kinds of paternity issues and things like that, because whenever there is money involved, there's paternity issues. There are paternity issues could be involved, and this testing in Jefferson's case went as far as when was he was one of the, I think he was the second president, I think, and some of these families were settled. Either they gave them property or monetary settlement, we don't know, but it's recorded in history. Now, coming to the Romanovs, Romanov was the last Tsar of Russia before the revolution. And why do we have Prince Philip's photograph here? That's because Alexandra was from the Queen Victoria's line. Now, so the Alexander Tsar, what was his name? So Prince Philip is the closest relative to the Romanov family. The Romanovs were all executed during at the start of the Russian Revolution, and all their bodies were taken and burnt out in the fields outside Ekaterinburg, which I believe is Petersburg right now. In any case, that was all that was left after the royal family was executed and the bodies were burnt. But then, when mitochondrial testing became a little bit more prevalent, they said the Tsarina Alexandra was actually a daughter of Victoria. So, and they said if they could connect the Tsarina's mitochondrial DNA to the remains of the rest of the bodies that were found, and mitochondrial DNA is more abundant than nuclear DNA, so they said there's a better chance of finding it. But what kind of DNA were they going to compare it to? So Prince Philip, and here is Queen Victoria, and then you have Alice, and Alice has Alexandra, and then the closest living relative to Queen Victoria is Prince Philip. So they actually got DNA from Prince Philip, tested his mitochondrial DNA, and then they checked the remains of the burnt and charred bodies from the woods in Russia. And they were actually able to show that both the Tsarina's DNA and the remains of the children who were also executed, they were all consistent with Prince Philip's mitochondrial DNA. Now, we are looking at historical investigations because this was very important to the Tsar's relatives. And the Tsar was not related to the was not related to Queen Victoria or the English royal family. So what they did was they compared his remains with another maternal relative on his side, and they were able to show that he and his great grandbrother or something like that. They were related because of mitochondrial DNA. And similarly, on the mother's side, the children were all related through the mitochondrial DNA from Victoria's line. And the woman who went around Europe and the US saying that she was Anastasia, when after she died, they found a piece of tumor material that they had taken out of her body when she had gone in for some medical condition. So they took DNA out of that tumor and they compared the mitochondrial DNA from the tumor to the mitochondrial DNA from Victoria's line, and they were able to show that she was an imposter. So this is all relatively recent history because they couldn't do this test until she was, Anastasia, she said she was not willing to give any DNA. So she didn't give any DNA, but they did get the DNA out of the hospital where she went for treatment. So these kind of things happen. These are all somewhat political. They are in the news quite a bit, but this just goes to show the power of DNA testing and also that how the technology has been used to resolve some of these age-old mysteries and questions. Now, just for 13 loci, the probability of finding a match is 1 in 600 trillion. So we can say if your DNA is clean and the profile is clean, then you have a pretty solid statistical basis on which you can say, I'm pretty certain that this DNA came from this person's body. So all this started in 1984 when Alec Jeffreys was a scientist in the UK, and he was playing around with what were called microsatellites, mini satellites at the time, and he came up with something called DNA fingerprinting. And somebody said, okay, you have this testing, and I will show you a picture of that. So there was an immigration case. They came to Dr. Jeffreys and said, there's a boy from Ghana and he's petitioning to become a UK citizen. And he says this lady is his mother and she's already a resident and a UK citizen. And how can we prove that the boy is not an imposter or the mother is not trying to get some other child in pretending to be the mother? So he said, let's look at this. And what he did was back in the old days, this was the level, we were talking about 1984, and these were the mini satellites. So they look at all the mini satellites. You don't know which chromosome it's from. It's just all the DNA from that person's body. And they just look at the repetitive sequences in the DNA and they say which one matches something. So if you look at all these bands here, you see that this band matches, you, you see two of these. This is the evidence here, and this is another band that matches. None of these others match. And so they were able to show that with similar results that the child was indeed the lady's son and he was granted immigration status. So this was done in 84, and this was the first time that they actually showed relationship using DNA to say that somebody was related to somebody. So that was the start of this. And then this was a very famous case called the Colin Pitchfork case. There was this guy who was running around the countryside in England, murdering and assaulting school children. And this microsatellite testing was actually used to show that the DNA that they recovered from the bodies of the victims was consistent with what the DNA that they got from this person called Colin Pitchfork. And that was the very first, to our knowledge, the very first forensic case that was solved using DNA. And that's also in 1986 and 87. And this was the state of DNA testing back in the 80s. We had something called restriction fragment length polymorphism, or RFLP. And this was one step ahead of the mini satellites that were used by Dr. Jeffreys, because you could actually tell which chromosome the samples came from. So if you can see here, Lane, okay, let me move lane 1 is consistent with lane 5. And the ladder runs in the middle, so you have the sizing. And you needed a lot of DNA to do this kind of testing, because this is when I got into DNA testing. You needed about 4 or 4 milligrams or micrograms, or how we explain in court. You need a size of a 50 paise coin, or a very large size of stain in order to be able to get enough DNA from it. So we are talking in amounts of grams here. So that was, and then you could see only 1 or 2 different loci, because now we are looking at 23 different loci at least when we do our testing. And here, using all this DNA, you can see maybe 2 or 3 loci. So coming to the next slide, now that we have the 23 loci that we do our amplification kits, how much DNA do we need to do this? Because we are amplifying all these loci at the same time. You're starting with the same amount of DNA, but it's all simultaneous amplification of all the 23 loci. And we are talking about a billionth of a gram. So we don't really need more than a nanogram of DNA, which is about a billionth of a gram, to get a decent DNA profile from a person's DNA after all that analysis, if it's from a single source. Of course, things get more complicated if there is more than one person um, contributing to that mixture, because mixtures are difficult to interpret. But if you have DNA from one source, and if it's a clean source, you don't need more than a billionth of a gram. So this is again how the technology has just improved and become more sensitive in trying to analyze the DNA that's available, and we don't have any choice in saying how good the quality of the DNA is. But the technology has definitely taken huge strides to help us with this. So these are 2 examples that I threw in there, which is not pertinent to this, but I just threw those examples in because the one was the case of a maternity. You have usually cases where there are paternity situations where, oops, okay. So there are cases when fatherhood is in question, but in this particular case, motherhood was in question. So this lady was a chimera, and she was making 2 kinds of ova. So the genetic disparities between her eggs were containing the eggs contained 2 different types of DNA samples, and this was also something that could be done only by genetic testing again, but they had to go into the medical records and things like that. So it's always important to not take things at face value, but also to question. When you're testing something, you need to know, because the first instinct that people had when they tested the kids was, oh, these 2 kids are yours, but these 2 kids are not yours, and you are an imposter. And why are you even saying that these 2 kids are yours? And she said, no, I gave birth to them. And then it turned out with genetic testing, it was shown that she carried 2 different types of DNA in her body. So DNA testing can be used to clarify situations like this. Pardon? So I'll try to. It's getting a little late here, so I'm kind of moving forward. So now mitochondrial DNA, and this is something else I want to point out here, because conventional wisdom is good. However, it's not all there is. So we believe that mitochondrial DNA can only be inherited from the mother, and this is just a cartoon that shows maternal transmission of mitochondrial DNA. Now, of course, to every rule there is an exception, and they found they found a case in Sweden. What happened was they found a boy who was having extreme fatigue situations. And when they tested his DNA, they found DNA from his father, mitochondrial DNA from the father in his muscle tissues. So while we say mitochondrial DNA can only be inherited from the mother, there can be cases, and we don't know how common this is. And this is the final slide. And this is the final slide. And there are lots of genealogy sites. You might have heard of ancestry.com and 23andMe, where people try to go and find out what kind of DNA they have in their families, especially people who are adopted want to know more about their biological parents. So that's becoming very common in this country. And I think it's getting more popular around the world. And now these sites are being used to do some kinds of familial searching. If you don't get DNA from the actual suspect in a case, they can actually go and see if there are your family members who have similar DNA, so they can close the window and they can actually go and interview people who might be possible suspects. And the last thing I wanted to say, one of the technological advances which can be scary and which can be a boon, is called DNA phenotyping. Just using some of the DNA, they can actually reconstruct what you look like. And they can have posters that say our suspect looks like this, or the missing person looks like this, if you have seen somebody like this. So these are all things that are available on the web, so I'm not going to spend too much time talking about, but these are all the trends that are becoming more and more common. DNA phenotyping was something that became very popular last year because they actually caught a serial murderer who was not known. He was very clever in concealing his whereabouts, and they were able to actually reconstruct a likeness of him just based on genetic data. And he was actually apprehended, and it turned out that he was the correct person. So a lot of different things are happening. And now these are the computer-based, the software-based kinds of technological trends that are getting a foothold, because the biochemical and the biological testing, they are already at a point where getting the data is not that difficult, but interpreting it is getting to be a little bit more challenging. So these are, when we talk about trends in biotechnology, there is a combination of the software as well as the actual hands-on work that is done with the actual samples and the testing. So that's all I have to say. I know we lost some time with the Zoom going down, but if you have any questions.
Thank you, ma'am. Thank you for your highly informative one. Dr. Madhu sir, is there
Thank you,
any
ma'am.
questions?
Yes, ma'am. Thank you very much, ma'am, for giving such a mind-blowing and entertaining presentation pertaining to the real-time issues, ma'am.
Thank you very much for giving me the opportunity.
Thanks a lot,
Now you
Abhi.
can start your video, ma'am.
Do we have questions?
Yes, ma'am. One more question from Shashikant. What bioinformatic tools can be used to extract mitochondrial DNA from raw whole genome sequence data?
You have to do because mitochondrial DNA is in the or mitochondria around in the cytoplasm. So you need other techniques, because the biggest challenge with mitochondrial DNA testing is contamination. I didn't go into that because we didn't have too much time, but so mitochondrial DNA labs have to be specially designed so there is absolutely no contamination. So in fact, a lot of labs choose not to do mitochondrial DNA testing and send it out to companies they specialize in that. So mixing the two would not be advisable, in my opinion, because mitochondrial DNA testing is it's a science on its own. It's it needs equipment which is slightly different. Even the air pressure flow in the lab has to be regulated to the exact to the um, exact limits in order to prevent contamination. Because when I was talking about drilling the bones, for example, drilling the bones or teeth to extract DNA, that we are looking for what do you call? We're looking for autosomal DNA, the whole genome. Now, you can, at that point, it may be possible to separate mitochondrial DNA from whole genome DNA, but I don't have, I don't, the techniques are different. And like I said, contamination is a huge problem. So that's the information I have.
Thank you, ma'am. Thank you very much, ma'am. I think that there are no more questions from the chat box.
Okay, but if anybody has any other information on that, because we have just gone through this dilemma for a long time, because we were building a new lab and they said, oh, if you're going to build a new lab, let's also get a mitochondrial lab. And when we looked at all the concerns about getting decent DNA data out of this, we said, okay, maybe we just stick to the whole genome DNA or the autosomal DNA, just because, like I said, contamination is a massive problem. But I can look it up and I can email. Any other questions?
That's all from this
Madhu
chat
sir,
box, ma'am. Yes, sir.
Madhu sir, there is a question in YouTube.
YouTube. Okay, sir. Please.
I'll tell that. How forensic science related to criminology and fingerprinting?
Yes.
If you throw some light over this, it will be useful. Is carbon dating methods also useful in forensic department? This is the question, ma'am.
Oh, yeah. See, forensic testing has so many different branches. So carbon dating is mostly for anthropology type investigations. Now, if it is important in a forensic case, it might be used. But it depends. Like I said, forensics is such an applied science. So it's always looking into getting techniques from all the other areas. And so is it useful? It depends on the case. I mean, any technique might be useful in forensics. That's my feeling on the whole thing. For example, stolen art. Somebody was asking me questions about how can you tell if a piece of art in a museum is a fake or real. Now, that's forensics too. So it depends on the case.
Ma'am, there is another question. How radiocarbon dating helping forensic, especially?
Oh, I haven't done it, so it depends. I mean, I don't know. I'm thinking of examples. Are we looking at, so somebody brings some fossil inside, somebody makes a fake fossil and says, okay, I found this while we were like digging. And so they want to find out if it's an authentic piece or not an authentic piece. Carbon dating might be able to resolve that. So it's case specific. We haven't come across forensic dating in any of our cases. Our cases are not that old that we have to use carbon dating for that. But there's always the possibility, like, it's anthropology is one of those things. Like, for example, there have been a lot of fraud cases when somebody says, oh, we dug these bones up and we found this and we think this is A, B, or C. And they say, we think this is what this is. And then historically, they'll say, when did that species exist? Can carbon dating show us some data that is consistent with this sample that was brought to us and the sample that it's supposed to be? So that is possibly one of the ways that you could look into whether carbon dating can be useful in this situation.
Thank you, ma'am.
Because you're looking at ancient samples there.
Yeah, thank you. Madhu sir, please take over.
Yeah. Okay, sir. That's all from the questionnaire, ma'am. Then thank you for your valuable speech and clearing all the doubts of the participants. Now I request my colleague, Dr. B. Prabhashila, to deliver vote of thanks.
Good morning to one and all present. I deem it a great honor and privilege to propose vote of thanks on behalf of the Department of Biotechnology. First of all, I would like to thank our guest speaker, Dr. Abhinami Chidambaram, for gracing today's webinar. Thank you, ma'am, for sharing your important work, your findings, and your opinion. Thank you very much, ma'am. It was very much interesting topic on forensic investigation. I owe my sincere gratitude to our Honorable Chancellor and Madam Chancellor for giving us this opportunity. We have been fortunate to be motivated by our vibrant Dean, Dr. Giri Rangasamy. Thank you, ma'am, for your constant support and encouragement in all our endeavors. I would like to express our profound gratitude to our campus director for his support and motivation. My sincere thanks to our principal, vice principals for their support, help, and guidance. I acknowledge the unwavering support from our HOD, Dr. Devika, in conducting this webinar. I'm happy to express my special thanks to all my faculty colleagues for their enormous cooperation in organizing this webinar successfully. We are very much thankful to Mr. Sam for his technical support and for his excellent coverage throughout this webinar. Finally, the wonderful participant who turned out in a great number. Thank you all for your enthusiastic participation. I once again thank everyone for their contribution has made this webinar series Trending Technology successful. Have a great day.
Thank you very much.
Thank you, ma'am.
Thank you, ma'am. Thank you for your presence and wonderful support.
Yeah, thank you. I wish it was not so rushed.
No.
We lost some time in between because I had a few more slides, but that's okay. Thanks
Thank you,
again.
ma'am. Thank you.
Abhi, all your sophisticated techniques were highly beyond my head, but still I could understand here and there. But it is very nice to find out that you are in the latest technology and you are doing good service to mankind to find out the proper victim and also the heir to the real heir to the property. So all these things are important because now everywhere we find malpractices. So you are doing really something good to mankind.
And thank you for being, and thank you, Giri, for being such an inspiring teacher, because, teachers are so important. And, we go back all the way to the 1970s.
Yeah.
So, but again, my teachers, my professors, and I mean, you are one of my first professors, and you kind of kindled that interest, and you want to push people further and see what you can do. And thank you so much for your inspiration.
Thank you. And it is late night for you, Abhi, whereas it's a bright morning for us. Hope
Yes.
we didn't disturb you.
Thank you,
that's
ma'am.
fine.
Good night, ma'am.
Thank you.
Thank you, ma'am.
Thanks.
Ma'am, shall I accept, ma'am?
Yes, sir. Yes. Thank you. Thank you, Sam. Thank you, Sam.
And thanks, Sam.
Yeah, ma'am. Thank you.